Okay, we're shifting from sickle cell to syphilis. It's my pleasure to introduce Sandra Ramirez. She did her undergrad or graduate studies in Colombia in microbiology, and then did her PhD studies in Spain, and then came to Ottawa, where she did a postdoc, and then became an adjunct professor in biochemistry, microbiology, infectious diseases, and also took on a role ultimately as senior scientist with Canadian Blood Services, where she knows everything about infections in blood. But today she's particularly talking about syphilis, I think. Is that right? Yes, great. Thanks, Sandra.
Good afternoon, and thank you, Ed, for your kind introduction and the organizing committee for selecting, this presentation for the symposium today. I would like to start acknowledging the people who actually have done the work. I am a messenger here. You can see Sylvia and Yunton. They are there in red, and the support of all the medical microbiology team, and in special Stephen Drews. We also have the support of other people from Canadian Blood Services like Mindy, Carlene, and Simon. This is really a team effort, and I want to acknowledge in special Caroline Cameron and Aloysius Gomez from the University of Victoria, because they provided the reagents that we needed to start this study, BI resources, and funding. We got a blood acceleration grant from Canadian Blood Services. So how it all started? I received this email from Dana in 2018. The two of us were heading to Colombia to attend a congress there. And actually, she was wondering if we could chat about syphilis testing. What she acknowledged is that there was no enough data by the FDA to eliminate this testing, and whether the bacterium that causes syphilis will survive during blood component storage, and if we wanted to do something about it. I do not remember the details. I know that we had dinner, but I don't know the details about the conversation on this topic. And after that, we did have a few meetings with NetCat and the development group, but nothing happened until 6 years later. We got this email from Mindy, and she had just read the March 24 issue of Vox Sanguinis, and there was a meta-analysis, and basically the conclusion was that there is no data to know if we should maintain or eliminate syphilis testing. And she was asking whether I had experience working with this bacterium, and I said no, but it was a challenge for me, and I really wanted to start working with the bacterium that causes syphilis, and it's Treponema pallidum. It's a microaerophilic gram-negative spirochete bacterium. It has 4 subspecies. The subspecies pallidum, so Treponema pallidum subspecies pallidum, is the one that causes the sexual transmitted disease syphilis, that is also transmitted through blood transfusion and transplantation and from mother to fetus. There are different stages of syphilis infections, so going from primary, secondary, and latent syphilis, and some people without treatment will develop tertiary syphilis. The latent phase can last 20, 30 years, and what is important here is to mention that people in primary syphilis before presenting symptoms, or people during the latent syphilis phase, they can be asymptomatic. And this is important for us because they could be blood donors. So, syphilis and transfusion medicine. I want to bring this paper by Stephen Drews and colleagues at Canadian Blood Services, what basically shows that, as we all know, there is an increase in the incidence of syphilis in the community. And this is not only in Canada, it's around the world. And this actually relates to the trend of an increase of positive donations for syphilis of first-time donors. However, what is important to notice here is that at repeat blood donors, we don't see that increase in positive testing. What do we do at Canadian Blood Services? We ask donors if they have been treated for syphilis in the last 12 months. We actually test 100% of donations, and if we have an initial positive, this is sent to different labs in Edmonton or Toronto for confirmatory testing. Then what is important here also to highlight is that these assays are not specific for the subspecies pallidum, that is the one that has been related to transfusion-transmitted syphilis. Positive test results including false positives, results in donor deferral, and this is actually quite important for us because of the test that we are doing, the cost that is involved, and basically that we are losing donors. And something that is also important for me to highlight is work done by Orton and collaborators, and it was published in 2002. And what they did, they took samples from donors that tested positive, confirmed donors, and they could not find DNA or RNA from bacteria there, from the spirochete. So the question is, okay, we can have positive donors, but are they really infections? And we know that donors can have, or people that have been infected with syphilis, even if they have been treated, they can have antibodies circulating for years, and it doesn't mean that they can be infectious. Other important facts, in Canada, there have not been transfusion-transmitted syphilis cases that have been documented, and worldwide, we have less than 10 cases after World War II. So really, there are no reports of transfusion-transmitted syphilis. However, the WHO still recommends universal testing. Despite these recommendations, countries like Denmark and Iceland, because of the very low incidence, they don't screen blood donors for syphilis anymore. And in Norway, they only test first-time donors. Other important facts is that there is no clear evidence of the role of leukoreduction in eliminating Treponema pallidum. This spirochete associates with platelets, so we really don't know if platelets will be carrying on or they can be filtered during leukoreduction. However, it's important to notice that at Canadian Blood Services, we have implemented the pathogen reduction technique intercept or treatment intercept of plasma and platelet concentrates, and there have been studies showing that this technology is effective in eliminating Treponema pallidum. What is the gold standard test? All of these studies that I have mentioned before, they test the viability of Treponema using a rabbit model. Especially, it can be a dermal test, or it can also be a model to develop orchitis or testicular inflammation. However, rabbits don't really represent the best model because rabbits don't develop secondary syphilis, for example, or tertiary syphilis. However, it has been the gold standard, and it's a very cumbersome test. So when we start talking about implementing something at Canadian Blood Services, and I was very keen on doing this, I was not keen on working with rabbits, to be honest. And looking at the literature, what we found is that the group by Stephen Norris, they have implemented an in vitro model that finally worked to be able to maintain Treponema pallidum in the lab, and it involves the co-culture of rabbit epithelial cells with Treponema in a very complex medium under microaerophilic conditions. So based on this evidence and the fact that this in vitro system existed, we decided to adopt the optimized in vitro cultures of Treponema pallidum to test treponemal survival during blood component storage. And based on this data, we would like to propose a reevaluation of the universal syphilis testing that we have right now at Canadian Blood Services. What is the study design? With the help, with not only the reagents, but also troubleshooting and communications with our colleagues from the University of Victoria, we were able to successfully establish the rabbit epithelial cell cultures. It seems simple, but it was not like that. And also, it was even more complex, establishing the in vitro culture of Treponema pallidum in these cells. We were successful, and in the interest of time, I won't go into the details, but it was not straightforward. However, we were able to do this. You can see here with dark field microscopy that we can observe the spirochetes. This has already been established by other labs. However, what is novel for us is to track Treponema pallidum during blood component storage, and this is what we wanted to do. We look at this paper published in 2021 by Tam Rackard, and actually Steve wrote an editorial about this paper. And what they did, they spike high load of treponemas in whole blood and platelets. And what they noticed is that the treponemas were viable. In whole blood, they were stored up to 7 days in the refrigeration. and in platelets up to 6 days in both room temperature or refrigeration. It was also viable in plasma for up to 28 hours. However, the limitations are that they, again, use a rabbit model. They spike with high loads of treponemas, and in platelet concentrates specifically, they did not use standard platelet storage containers. So, what Sylvia and Yunton have been doing in the lab, they grew treponema in the rabbit epithelial cells, treated these cultures with trypsin and rinsed the cells, and used this to inoculate platelet concentrates. Platelet concentrates were stored in either transfer packs that there are no oxygen permeable, or standard platelet storage containers. These were stored for 20 to 24 degrees for up to 7 days, and samples were taken at 5 and 7 days. And we thought that we were very optimistic that we will be able to just take a sample under microscopy and observe treponemas. But it was quite difficult because there's a lot of platelet debris, and we cannot see the treponemas very well with dark field microscopy. So we were thinking that to test the viability of treponemas after platelets had been stored, ideally what we want is to reinfect fresh epithelial cells, and that's what we did. So we took samples from platelets, infected rabbit epithelial cells, and then we harvest treponema pallidum, first of all for dark field microscopy, and eventually we also want to optimize immunofluorescence and PCR assays. And this is what we did. Unfortunately, I had a short video here and it did not work, but just believe me, these treponemas are viable, and the way that we know they are viable because they have the typical movement. And you can see also that there is very high load, because to be able to see this under dark field microscopy, you should have 10 to the 6, 10 to the 7 treponemas per milliliter. So in conclusion, we know that treponema actually survives in platelet concentrates stored up to 7 days in both type of containers, gas permeable. We are surprised about that because treponema is microaerophilic, and also in non-permeable conditions. Then the next step was obviously to move to red blood cell concentrate, that we know that is the product that is mostly transfused. We had the same approach. We grew treponema in the epithelial cells, treated these cultures or samples with trypsin and rinsed them, and we spiked red blood cells. Initially, we also wanted to see if we could observe treponemas there, but it was not possible due to a lot of background noise. And to our surprise, when we had the same approach, or we took the same approach that we used with platelets, we took a sample from the inoculated red blood cells into epithelial cells, and everything clot. It just forms a clot. And we're thinking, so what is happening here? So there were a lot of troubleshooting. And what Sylvia found out is that just red blood cells without treponemas, and the medium that is used to grow treponema in the epithelial cells, this is what actually causes that clotting in the red blood cells. It's not the interactions between red blood cells and epithelial cells. That's where we are right now. Here, we know that we inoculate 5 milliliters into 45, so it's 1 to 10, and we don't see that coagulation. However, when we take 5 milliliters into 5, we do see that coagulation. So we presented this data to the medical microbiology team, and we got some ideas for troubleshooting. Again, what we think is that there is a reaction between the red blood cell additive solution and the medium that we use to grow treponema. And you can see the composition of the medium. It's very complex, and it has 20% of fetal bovine serum. It's not only this. We have to buy this reagent specifically from these vendors, and it has to be this catalog number. So this has been optimized by Stephen Norris' lab and also at the University of Victoria. If you change anything here, treponema does not grow. So that's the challenge that we have. Right now, we are looking into different approaches. We would like to dilute the sample with plasma. That's something that we discuss, or wash red blood cells, or lyse red blood cells, or filtering, try to recover treponemas. We'll get there, but right now this is what we are doing to troubleshoot the coagulation that we observed. Other things that we are doing, we want to see if treponema basically survives under refrigeration. And for that, we will be doing experiments with cold stored platelet concentrates. And something that is very important is that, again, all of these experiments have been done with very high loads of treponemas, 10 to the 6. And we know that less than 10 treponemas are infections in humans. So what we wanted to do is to titrate down until we see what really is the load of treponemas that will survive during blood component storage and can be infections in epithelial cells. We want to optimize immunofluorescence assays, and we have already developed certain reagents to detect virulence factors from treponema. At the end of the day, this is very interesting, this is research, but we would like to bring change to practice. So the idea will be to propose approaches to maintain or modify our current donor testing for syphilis at Canadian Blood Services. The take-home message, we are the second laboratory in Canada that has been able to establish an in vitro culture of Treponema pallidum, and we are quite proud about that. We do know that Treponema pallidum survives in platelet concentrates that are stored up to 7 days. However, right now we have implemented pathogen inactivation of platelets, so that's not an issue anymore. However, these technologies that now exist for red blood cells, and we would like to be able to solve the challenges that we have right now, so it's a work in progress. And with that, I would like to thank you for your attention. We'll be happy to answer any questions.
Sandra, that's great. Maybe you could just educate the audience, just explain why screening for syphilis is so important.
Well, it was important since the beginning because before the Second World War, we knew that there were transfusion-transmitted syphilis cases. But the other thing is, for years, testing for syphilis had been used as a surrogate test for HIV. And now, based on studies done by different people, but one publication from Canadian Blood Services, Sheila O'Brien in 2023, she showed that despite that the incidence of syphilis is increasing in the community, the incidence of HIV is not increasing. So even the value that was posed at one point of time about testing for syphilis to decide if donors may also be infected by HIV is not valid anymore. So that was basically why we implemented that.
The other key problem was that the antibody tests we're using can be stable for the donor's lifetime. So you can have a donor who's been treated, who changes their lifestyle risks, who's not infectious and now is deferred indefinitely. And so we wanted to see whether we could move around that with getting some data on survivability in products as well.
Hi, Sandra. I'm not sure if I'm actually correct on this, but didn't the incidence of syphilis really drop, and then kind of made a resurgence? Or did
actually, in the last few decades, the incidence has increased dramatically. We're talking about like a 1,000%. But and that trend is similar to first-time donors, but not to repeat donors. Yeah. So in the community, and for us, for example, we see it's like a 6.6 per 100,000. But in other countries, it can be 2%, and even in some countries, it can be 8% of first-time donors testing positive for syphilis. Yeah.
So we actually see a massive outbreak in Western Canada. In particular, Alberta, we've seen that. I was in public health there in 2009, and we started to see a massive spike. With that spike, you get expansion into sexual networks where people are unaware of their risks, so you get a massive increase there. The data that Sandra showed, we've seen just general infectious syphilis rates increase. We've seen vertical transmission, and even in the middle of the pandemic in Alberta, that were the highest rates we'd ever seen for years. Rates that were higher than before penicillin was introduced. And then the other thing that we've also seen, though, is that when we look at our donors and we look at their RPR levels, which are kind of a surrogate for infectivity or not, and they're not always the greatest, we do know that at least our donors, when they come in, are generally they have no RPR levels or very low levels, so probably not infectious. So they're probably reduced, risk managed down. But once they're positive, and you can't, you could try to bring them back, but the return on is quite low because the tests we use now, they're going to be positive by serology for life.
Yeah, and the incidence, when we see that increase in incidence, a specific population with high risk of behavior and specific ethnic groups and social conditions as well. Yeah. So.
When you finish this study, Sandra, and you find out that we don't need to test for syphilis, how much will you be saving Canadian taxpayers?
Well, I really don't know right now what is the cost of the test, Steve. Do you have any idea? Because there are two approaches. We either eliminate tests, but what I'm thinking is that probably I will be more comfortable with doing something that what Norway is doing, testing only first-time donors. Because first-time donors, actually, we also see an increase in the incidence, but I do not know what is the cost of a test. So.
It wouldn't just be the cost, but because it's so many donors going through the system, you could open up another channel for another test. Like we would, the cost can vary, but it would be a cost savings, and then you could have space to do another test in your workflow.
Steve, ballpark. Can you?
Yeah, anywhere from $3 to $10 a test, and you're getting 900. You're getting like 800 to 900,000 donations a year, and everyone gets a test. So, yeah, you're saving a significant amount. And then workflow, the workflow is a big one, right? Because it's like you got to do that on everything. So if I open that channel up, I can test a donor for something else.
We could put all that into grant money.
You could.
Of course, yeah, good.